Cancer is one of the largest problems in medicine today, claiming millions of lives. You may have had a relative or friend who has suffered from it. But how does a normal cell turn into a cancerous cell to start with? It turns out it's a fascinating journey involving the core of life, the DNA. DNA is the building blocks of life. At the moment, millions of your DNA strands are reproducing. How does this work and how does it lead to cancer? DNA is essentially a double-strand nucleotide biomolecule which contains a bunch of genetic instructions and is essential for human life. DNA could be considered a database or storing all the information, though not responsible for execution. Note that this is different from RNA. RNA, standing for ribonucleic acid, is responsible for executing the instructions and facilitating the output, typically in the form of proteins. So how does DNA work? DNA is made up of three sections: a phosphate, a sugar, a nitrogenous base. In a nutshell, these nitrogenous bases are different and consist of adenine, thymine, guanine, and cytosine. These instructions will be carried through RNA to typically make proteins to carry specific functions. The key problem occurs in DNA division during mitosis. Mitosis is a process where cells and chromosomes divide. Usually, mitosis occurs without flaws, and crucially, the chromosomes of DNA stays intact. However, mitosis occurs thousands of times and slowly mutations build up. There are thousands of mutated cells in your body right now, though they're not considered cancer. However, these usually don't become a problem due to the cell cycle. The cell cycle is a repeating process where a cell grows and develops before mitosis. Other than mitosis, cell imperfections can still occur, so there are various timings where proteins can inhibit mutated cells. One of these proteins is p53 and is responsible for triggering apoptosis or cell cycle arrest, a limbo cell state where cells go into during DNA repair. However, there are loopholes. All proteins manufactured to inhibit a cell are produced by the cells itself. When the DNA used to synthesize inhibitor proteins is corrupted, the inhibitor proteins are not synthesized, forcing the cell past no return. How it turns to cancer. At this point, the cell is permanently abnormal, though not cancerous. The final mutation occurs when the oncogenes get corrupted, causing massive production. This is now a cancer tumor and could potentially be deadly, though not certain. In summary, DNA corruption leads to various mutations which can make cells abnormal or even cancerous, however need to overcome various mitigating stages. While it can occur to chance, living a sattvic lifestyle and staying off additives can lower these chances. With the help of medicine discovery, we may be able to defeat cancer into a thing of the past.